right, I'm going to go ahead and get us started for today. So, it is my distinct pleasure to welcome Anthony today to ML for Proteins. Anthony did his bachelor's and his master's in the life sciences and technology at EPFL. He also did his PhD there in bioengineering and biotechnology with a specialization in computational protein design and engineering. He even won Best Thesis for this. He worked in 9 different labs in academia and industry throughout his time so far, and he's currently working as an R&D scientist across the road at B Novate Technologies for the development of biochemical reagents for the automated detection of specific bacteria in water. Today, he's actually going to be talking about his recent Nature paper targeting protein ligand neosurfaces with a generalizable deep learning tool. So, Anthony, thank you so much for coming today. We are so excited. So please take it away whenever you're ready.
All right, so yeah, thank you for hosting me today for this talk and to present to you the recent advances that we proposed in Nature, as it was explained. And today we're going to talk about how we can use machine learning and a concept that we call neosurfaces in order to design novel protein ligand interactions. So from my background, I'm not like a machine learning in machine learning, but I will try to explain as much as possible as I can. And the goal today will be to show you some translational application of how we can use machine learning tools to design protein binders that can then fulfill a therapeutic function. And actually, designing protein-protein interaction, also abbreviated PPIs, is not as trivial as fitting 2 pieces of a puzzle because they are like very complex. And there are 2 questions that you need to solve if you want to design a binder against a certain protein target of interest, which is where do you want to bind on your protein target and what do you want to bind to it? And this sounds like very trivial, but actually this is very complex because proteins are not just like 2 geometric pieces that you fit together. You also have to take into account the chemistry and so on. So that's why it's very complex for just human being just to assemble together. And therefore, I will like proposed a geometric deep learning tool like 4 or 5 years ago, which is called MaSIF, which stands for Molecular Surface Interaction Fingerprinting. And what MaSIF does is to look at the molecular surface of the protein. So it's kind of the skin of the protein. You have the skeleton inside and then you have the skin. And you try to look at the different geometric and chemical features that you have on the surface of this protein. And among the geometric features, you can have some knobs, pockets, so you have different curvature. You can also have different chemistry, like an hydrophobic index, proton donors, proton acceptors, but also different charges in terms of electrostatics. And you can use these different features that you have in the space. You can vectorize these features, and then you can feed a geometric deep learning algorithm to learn about them and to compute what we call the descriptor, which is basically a vector that describes the protein surface. And we usually like to compare these descriptors to a fingerprint because all of you have fingerprints that are very specific to yourself and tell about your identity. And so does a descriptor for a certain protein patch on the protein surface. And we try to train these different descriptors to solve different tasks. One of them was to identify on the protein surface which site has a highest propensity to be involved in a protein-protein interaction and which one is not. And the other one was massive search. So it was an all against all search algorithm in order to match and dock each protein partner together. And with these 2 tools, we kind of answered the where and the what question that I mentioned before. And MaSIF was very fast and accurate to do so with known protein-protein interaction that we have from protein databases. And recently, also in Nature, like 2 years ago, we also published an optimized version of this tool. So we tried to leverage this massive site and massive search, and we created MaSIF-seed to design novel protein interaction for different target of therapeutic interest. So I don't want to go into too much detail because basically what I'm going to talk today is using the same architecture and the same strategy. But just to show you what we achieved so far, was actually to do protein interaction. For example, here for SARS-CoV-2, so the RBD domain of SARS-CoV-2, so it was in 2020 when we initiated this project. So it was right at the beginning of the pandemic. And we also had the binder for PDL1, which is a protein which is aberrantly expressed on tumor cells. And we also had the binder for PD1, which is on the other side of PDL1, which is found on T cells. And we found binders for all these different targets having up to 80, 65, and a few hundred nanomolar of affinity. And they were all confirmed with either structural data or mutational characterization. So overall, it was quite successful to design novel protein-protein interaction from a computer by using this approach. And then we thought, okay, maybe we can also now involve small molecule in that process to have, for instance, a protein-protein interaction that is conditioned to the presence of a small molecule in between and will bring these two proteins together. And MaSIF is actually working with these different geometric and chemical features that I mentioned before. So just to recall you, there is the curvature and the geometry, the hydrophobicity, the proton donors and acceptors, and the electrostatics. So that's the main features MaSIF is working with. But initially, this was only trained and used with amino acids. So we had to make a few adjustments in our pipeline so that now these different features are also computed for the small molecules. And with that, the rest of the job was basically the same as before. So we can also do an interface prediction in order to see if the interface propensity is changing upon addition of the small molecule. And then we can also make a search like we were doing before and try to see if now, once the small molecule is there, the two protein components are coming together. And the fact that MaSIF can actually generalize to small molecules is basically that it does not care what is underneath the surface. It just cares about what kind of geometric and chemical features I have on the surface. And it doesn't care if underneath you have an amino acid or any kind of other ligands. So all this interface prediction and partner search were exactly the same as we were doing previously. And then the idea was to then make de novo protein ligand interaction, which are conditioned to the presence of the small molecule. And to do so, you take an existing protein ligand interaction. You have here BCL2 in complex with venetoclax, which is a clinically approved small molecule for leukemia. And then you ask MaSIF, where do I have more chance on this complex to have a protein interaction happening? And then you extract a patch and you extract the fingerprint of that patch. And then you try to look on database that we generated if we can find a complementary fingerprint in our database. So we took lots of different proteins, we chopped them in different helical and beta strand pieces, and we compute a fingerprint database. And then we try to see if we can find a complementary piece of the puzzle in that database. And to do so, we take our fingerprint, we invert it, and we look for the closest neighbors around this inversion. So the idea is to find the complementary piece of the puzzle. So if you have a positive charge on one side, you want a negative charge on the other. If you have a proton donor, you want a proton acceptor, and so on. So you invert your fingerprint, you find the closest neighbors, and these different complementary patches are associated to a small protein fragment that we call the seed. And this is called the seed because that's the starting point around which you will design your novel protein interaction. And then you have different scores and metrics to select the seeds given by MaSIF. And then this seed is then docked on the protein surface of your protein ligand complex of interest. And because we're not simply working with peptides, we need to refine this peptide and stabilize it on a scaffold protein. So we have a refinement step first, which is done with Rosetta. And the idea behind that is that MaSIF will give you the best seed it finds in the database, but it's not necessarily the most ideal seed ever. So you may have some little clashes or an H bond that is not optimal. So this little adjustment with Rosetta can actually give you a better seed. And then we are grafting this seed on a protein that we call the scaffold. So that scaffold will stabilize the motif in that conformation, but also bring some additional contact on the rim of the interaction in order to get even better binding interaction. And again, this process is done with Rosetta and also with an energy optimization process. So once we had this pipeline, we tried to see if we can validate this approach in vitro with different protein ligand complexes for which we design a de novo binder. And we had 3 test cases in mind. They're like very versatile, very different. We have BCL2, which is a pro-apoptotic protein in complex with venetoclax. So as I said before, it's an FDA approved therapy against leukemia. We have progesterone, which is a hormone which is bound here to an antibody that recognizes progesterone. And we had actinonin bound to PDF1. So PDF1 is a peptide deformylase from Pseudomonas aeruginosa, so it's a bacteria. And we have actinonin, which is an antibiotic. So like very versatile molecule coming from different origins. And then we asked MaSIF what it thinks about these different protein ligand complexes in terms of chance to design a novel protein interaction. We had like very modest to quite high interface prediction, which were predicted by MaSIF. We selected different patch for each of these protein ligand complexes, and we tried to design de novo protein binders based on the pipeline that I just mentioned on the slide before. And with this, we designed 2,000 designs for each of these protein ligand complexes, and we tried to make a high throughput screening in vitro using yeast display to screen for these different binders and find some hits. And I guess not all of you are familiar with yeast display, so here I have a very small recap of yeast display and how it works. So it's basically a way to avoid to make an expression purification of every single protein you want to test. Here you actually pull all your sequences together and you transform genetically some yeast that are then sorted by flow cytometry in order to find some hits. And the way it works is that you genetically modify your yeast in order to express your design of interest on the surface of the yeast. And then you can come with your target, which is directly or indirectly fluorescently labeled. And this way you can recognize the yeast that bind to your target of interest. And we also have a tag on the C-terminus of the yeast so that we can check if the design is properly expressed or not. And this is what you see here on this scatter plot from flow cytometry. You have on the x-axis here the expression tag, just to check that the design is expressed. And on the y-axis, you have the target binding to the design. And what you want to sort is actually the double positive, so the yeast that express and bind to your target protein. And you do that for several rounds. So you have a first round where you get a little bit of binding. You take that population, you put it in culture, and then you do that one more time. So you do one more sort, and now you see that you start to get some binding signal being enriched. And then you sort the binding and the non-binding population. You go to deep sequencing, so genomic sequencing, and you try to see how many times each design is in a binding population versus in a non-binding population. And this way, you can check which design were properly enriched in that process and which one are the winner of the competition. And at the end of this process, of this selection process, we ended up with one design for each protein ligand complex. And as you can see, they have different topologies, different binding modes. And most importantly, they were binding only in the presence of the small molecule. You see here the flow cytometry data of this clone. So this is the yeast clone that displayed this very specific design and labeled with this target in presence or not of the small molecule. And you see that if the small molecule is absent, we do not have any binding signal. This is the histogram that you have here in red. And once we add the small molecule, we do see a shift in the binding signal. This is the blue one. And this actually tells us that the protein-protein interaction is only happening once we add the small molecule. So this way, we designed the on-switch system that we wanted. And now the question could be, okay, but how are you sure that this design is binding to the right place? And to do so, you have different methods to check that the binding mode is correct. You can do a knockout mutants. So basically you take the interface, like here, for instance, and you see that this methionine is at the core of the interface. So if you mutate this methionine to, let's say, an arginine, for example, which is charged and very bulky, if you are at the right place, this should not bind anymore. And this is what we do here. You have the design, the negative control, and the design with one single point mutation at the predicted interface. And now we see that there is no binding. So we are quite sure that this design was binding to the right place. You can also change the drug. So if you have a drug analog, which is the case for BCL2, in principle, this should not bind if you just change the small molecule. So here, we change venetoclax here in blue to this other analog binding to the same spot. And you see now that we do not have any binding. So we were also quite confident that we were binding on the right place on BCL2 itself. Or the last one, which is, in my opinion, the most elegant one, is called the site saturation mutagenesis. And the way that it works is that now you take your design and you do another screening by this display, but this time each yeast will display a single point mutation of your design. So you take all amino acid position and you mutate them one by one to all possible amino acid, and it will give you this 2D matrix, and then you will get a heat map, and the color corresponds to how many times do I found this mutation in the binding population versus non-binding. So the more blue, the more beneficial this mutation is, the more red, the more deleterious that is. And you see that the regions that are here very red are actually the ones that are found at the predicted interface. And if you want, you can even map that to the original protein ligand prediction that we had initially on the computer. So you do the average per position of this different mutation, and you report that mean sensitivity on the PDB that we initially constructed. And here you see that the regions that are in red are the ones that are found at the interface. So overall, with this process, we're quite sure, before doing any crystallography, that the binding mode is quite correct. And another matrix that is very important for any protein designer is, okay, now it binds to the correct place, but how strong? And we did that in presence and absence of the small molecule for all of them. We had like almost no binding or very weak binding in absence of the small molecule. And we start to get like very decent binder displaying half a micromolar to about 10 micromolar of affinity. This is still a weak interaction, but considering the difficulty of the task of designing protein ligand complexes, this is still a very good binding, which is still in a range of native protein-protein interaction. But by using the site saturation mutagenesis data that I just showed on the last slide here, you can get a hints on how to improve the design. For instance, here you have a very dark blue spot, which suggests that this mutation here could be highly beneficial for the binding. And we identified 2 mutations in each of these different protein ligand complexes. Some of them just reduce a bit the entropy, like replacing a methionine to a leucine. So a methionine is very entropic with a lot of degrees of freedom. So you can easily reduce the entropy, but keeping the hydrophobicity by replacing it to leucine. So this is one of the things that we are learning from that process. We had a little clash that was not really seen on the computer initially. We had a better hydrogen bond that was done here. Here we have a better pi-pi interaction and pi-pi stacking, and here another hydrogen bond. So overall, there are different things that we didn't really see on the computer that we are now noticing once we do the experimental validation. And we can use these different mutations that were obtained from in vitro validation, and by introducing them in the design, we improve the affinity from 100 to 1000-fold. So the most impressive one is maybe the progesterone binders, this DB-Pro 1156, that was above 10 micromolar before, and now we're like around 18 nanomolar, just by doing 2 mutations. We had about 100-fold of improvement for venetoclax and like very slight improvement for actinonin, which was already quite good before. So overall, just by doing 2 mutations, you can go from very weak binding to very strong binding. So every single mutation matters. And as I said before, the ultimate assay for any protein designer to check that what you want from the computer is really what you get in reality is the experimental validation and the, for instance, crystallography or cryo-electron microscopy. And we did cryo-EM for the progesterone binder, and you see it here. So you see the overlap between the computational prediction and the experimental data. We had a little bit of offset, but still the binding mode is correct and the amino acid are placed properly. But this flexibility could be due maybe to the CDRs of the antibody, which are very loopy and very flexible. So this was already hard to model. And the other one, the actinonin binder, is binding almost perfectly. So we had a very good agreement between our expectation and the experimental data. Unfortunately, we didn't get any crystal for the BCL2 venetoclax binder. This didn't crystallize, but considering all the experimental data that I showed before in terms of knockouts, saturation, mutagenesis, and small molecule analog, we're very confident that the computational prediction are the same in reality, despite the fact that we didn't get a crystal, unfortunately. And then some people may say, okay, but you didn't really use AlphaFold in that process, and it seems that AlphaFold is nowadays a golden standard. And so what does AlphaFold think? Well, when we initiated that project, AlphaFold 3 was not available, it was not released, so we could not incorporate small molecule in that process. Still, we can now analyze the binder itself as a monomer, not in complex with the small molecule and the target protein. And what we noticed once we had the crystal was that there was a little bit of offsets at the N-terminus of this actinonin binder. And if you look at the amino acids that is involved in that process, this tyrosine at the beginning of the protein is slightly off, which then could explain potentially why we have this little divergence between the computational prediction and the reality. So this is what we get between the crystal and our initial model. And if we had run AlphaFold from the beginning, AlphaFold would have placed it like perfectly at the right place, despite the fact that AlphaFold has never seen before this de novo protein design. So this was actually quite remarkable that AlphaFold can be very accurate with de novo protein design that were not part of the training data. And then we thought, okay, maybe we can learn from that and improve our pipeline, incorporate AlphaFold to try to save and rescue the bad designs. Because if some of this design, because we tested 2,000 design initially, but only a couple of them bound. If a design is not binding, there may be two reasons. The first one is that the monomer itself is not folded properly, or the other one, which is a bit harder, is that it's folded properly, but it just don't do the job. So the second one is harder to predict, especially that we didn't have AlphaFold 3 at that time, but using AlphaFold 2, we could already see if the monomer were properly folded or not. So this is for each protein ligand complex, the AlphaFold prediction of the monomers of the de novo designs. And you can see that a lot of them are not predicted as folded by AlphaFold. But the one that we validated before, so the orange dots, were all predicted as folded. So we tried to use ligand and PNN. So I think it just has been published in Nature like a few days ago, but the preprint was already released in the end of 2023, if I'm not mistaken. And we actually use ligand and PNN to improve the design and make sure that now it has a better folding. And this is now the success of folding predicted by AlphaFold after the optimization with ligand and PNN. So we have now about 80% of our designs that were predicted as folded, while before it was more about 30% of them that were predicted as folded. So it was a huge improvement in silico, and we tried to test them again on Yeast display. And now we had a few more hits. This is the dark dots that you see here and there. And these designs before were just not seen because they were not predicted as folded for most of them. And this is the additional designs that we brought for either the actinonin PDF1 complex and for BCL2. We didn't have any other for the progesterone binder, unfortunately, but still we see now that we have lots of different designs that share some common seed for some of them, but overall they are quite different in terms of topology, binding mode, and so on. And this is the improvement of the success rate. So initially we had like only one binder out of 2000, which is not a very great success rate, but with ligand and PNN and the alpha-fold prediction to actually filter the bad designs from the good designs, we had a success rate that reached up to 2.5% in the case of the actinonin PDF1 complex. And nowadays, now that we have tools for PPI design like Minecraft, for instance, or RF diffusion, this may sound like very low because nowadays you can still reach between 10 and 50% success rate depending on the tool. But considering that we have a small molecule involved in that process, the success rate is still remarkable. And then what can we do with this? Because we just designed protein ligand complexes that are conditioned to the presence of the small molecule, but what is it useful for? We try to make some translational application in cell-based therapies. One of them was to fuse our progesterone binding system to an RNA polymerase and a zinc finger so that we can have an inducible transcription of a gene that then is translated into an mCherry. We had another system which is called GEMS. So this is generalized extracellular module for signaling. And basically here, what you do is that you have a receptor that change conformation about dimerization. And this change of conformation will then trigger a STAT3 pathway and the expression of a reporter gene. We had another assay, which is basically to have a split non-luciferase and our two components of the PDF1 actinonin system. And once they are brought together with actinonin, we have then we can then trigger luminescence. So this can actually be a nice example of a biosensor. And the last one, which is maybe the most interesting one in terms of translational application, is actually to have a switchable CAR T cell, which is the one that I'm going to explain a bit deeper in the next slide. So I'm not sure that all of you are familiar with T cells, so just a very small recap CAR T cells are actually immune cells that we take from the patient, and they are genetically engineered and reprogrammed so that they can recognize the cancer cells and be more effective. They are then expanded and re-injected into the patient. They are called CAR T because it's called chimeric antigen receptor T cell. And basically, we just give a gene that will redirect the T cell towards the cancer cells of interest. The job basically is that this T cell will attack the cancer cells, but not the healthy cells. But this is not always the case. So you may have some off-target activity, and on the other hand, you can also get some exhaustion because the T cells is too active and it's not effective anymore. So what would be very interesting is to have a control CAR T cell that is active only on a certain time window. So basically, it's in the dormant and sleeping states by default. But once you give a drug or small molecule, it becomes active and can kill the cancer cells, so that you avoid exhaustion and you will avoid some off-target activity outside of this therapeutic window. And to do so, we actually took the classical CAR of CAR T cells. So normally you have a recognition domain on the extracellular side and a signaling domain on the intracellular side. And here we split these two domains into two, and we introduce in between our venetoclax inducible system. So we have BCL2, which is fused to the recognition domain, and we have our venetoclax binding protein, which is then fused to the signaling domain. And this recognition and signaling domain come in close proximity only if the small molecule is added. And this is what we have seen in reality as well. So you see here in the middle, the classical CAR with or without venetoclax and the killing activity, which is reported here. And the killing was the same for the classical CAR, independently of the presence or not of venetoclax. So it was just killing. But for the inducible CAR that we designed, we didn't have any killing or very residual killing in absence of the drug. And then we had an inducible killing activity that we observed in presence of venetoclax. So we can really turn on and off our T cell upon demand, which is exactly what we wanted to show here. So now if I have to briefly bring a conclusion, so in this work, we actually showed that the surface fingerprinting approach is efficient for protein-ligand interaction design, but also that it was very easy for us to bring the small molecule in that process because MaSIF is easily generalizable to components that are not amino acid. Because MaSIF doesn't occur if you have an alanine or valine, it just cares what kind of features I have on the molecular surface of my protein. And these geometric and chemical features that we use are common to both the small molecule and to amino acid. So that's why actually this tool is easily generalizable. And in that work, we also show that we could indeed design protein binders that are conditioned to the presence of the small molecule. And this way we can design novel on-switch systems. And these different on-switch systems were then functionalized for therapeutic application like biosensors and also in CAR T-cell therapy, like I just showed before. And hopefully this can be used in the future for different kind of application in terms of synthetic biology and for many different bioengineering applications. So with that, I have to thank like many people because this work has been done like for the last 4 or 5 years. So we had this first Nature paper with the MaSIF-seed pipeline. This is most of the collaborators that participated in the first MaSIF project. And there were also like lots of different people that were involved for the MaSIF neosurf in terms of translational in the lab for cell-based application and so on. So I have to thank all these people and especially my supervisor, Bruno Correa at TPFL, and of course, all the co-authors of this paper. So I'm happy to answer any question if you have any. Thank you for your attention.
Thank you so much, Anthony. I will give it a minute to see if anyone has questions. You are allowed to unmute yourself at this point if you would like to ask a question that way, or feel free to put it in the chat and we can read it out to Anthony and he can answer it. I might try to get us started with a question. So I've seen a lot of research recently for drug binding and design around things that don't have like a full structure, whether it's not necessarily an IDR, or yeah, maybe it has like some structure, but it's not complete. So do you see that fitting into your pipeline if someone were to just do their best to like have a homology model or predict something with AlphaFold and then use your model to try to design around it? Do you think that those types of methods would work here with your current pipeline, or is that something that you really need like a solid structure for?
So in theory, it could. There is no objection against that. But the little problem that we have is that with such disorder structures that you have a lot of flexibility. So MaSIF is only working with a rigid conformation, so a snapshot at a certain conformation at a certain time. So if the input that you give is not representative of the conformation that you get in reality, you will be undersampling the different possibilities. So one idea could be to have like a molecular dynamic simulation that brings you different kind of conformation, and then you run MaSIF in parallel for all these different conformations. So that's one idea. But overall, most of this protein-protein interaction design that has been done today and in the past work better for rigid structure because, well, if you do not have, if you don't take into account the flexibility and the entropy that it generates, you get a higher success rate. And most of the papers in the novo protein design that were done in the past that were quite successful, they were done on very rigid interface. And I guess it would be the same here.
Gotcha. Thank you so much. We had a question in the chat that asked, could you introduce some information about the data set that you used for your model?
So if you mean for the training of MaSIF, so this was done like a while ago. So there were different steps. So we used different data from the PDB bind back in the days. I think it was like much better now, and other PPI database that I do not remember by heart. So if you look at the Nature paper that we have like very long time ago in 2020, you will see the name of the different database that we use to generate the training set. And then for the MaSIF-seed, so for the seed that we use, this one came from the PDB, and then we just chop them in different motifs. And then for the scaffold protein that we use for grafting the seeds, this scaffold come either from the PDB or from some mini protein from the Becker lab, two mini protein paper from the Becker lab, and we also had one or two from Eva Maria Strauss lab as well.
Awesome. William asks in the chat, has your team tried D-MaSIF and compared the performances of these two models? D-MaSIF costs much less computational resources than MaSIF.
That's true. It consumes much less. The problem is that it was never benchmarked for experimental validation before. And well, it would have been a bit too risky at that stage of the project to switch to DMassive. So that's why we kept it as it is right now with the regular MaSIF. But of course, it will be much faster and consuming less resources to go with the DMassive. But the good thing actually with the regular MaSIF is that you can make easily a parallelization with CPU, so you don't need to get a lot of GPU like you need for DMaSIF, for instance. So here you can easily parallelize with CPU, so this was not really a big problem for us. The only problem is basically the C database, which is huge because you need to pre-compute all this data before. And well, that's the only limitation that I see, but otherwise it performs well.
Awesome. Another question from the chat asks, could you comment on how similar or different the fingerprints are for closely related or homologous proteins? Could there be off-target effects if fingerprints are similar?
I guess they could. I mean, of course, it depends on the amino acid that you have on the surface because that's what actually dictates the geometric and chemical features that you will get. So if one of the mutation is an arginine switching to an aspartic acid, for instance, you will change the charge. And here, maybe this is enough not to have a protein interaction happening. Well, we have to see that in reality, but for sure, if the sequence is conserved or quite conserved in terms of homology, we will for sure have similar fingerprints as well, because the fingerprint is dictated by the features, and the features are dictated by the sequence.
Awesome. Anna asks, sorry if I missed this, but what design pipeline did you use to design the binders?
Yeah, well, this was at the very beginning. Sorry if I was too fast. Yeah, here we go. Yeah, so if I just recall it in a minute, so basically the pipeline was the following. So we had our protein ligand complex, and then we asked MaSIF which region has the highest propensity to be involved in a protein interaction. And then MaSIF is looking for a complementary fingerprint. So all of these steps are done with MaSIF. So MaSIF tells you the place and the binding motif that can go at that spot. And then it will dock it on the protein surface. So for that, it's using an algorithm called RANSAC. So it just take the descriptor at 3 different spots, and then it just make an alignment to make minimization. And then the last two parts are done with Rosetta. So here we have a seed refinement step. So it's using a fast design protocol on Rosetta with an emphasis on building a better hydrogen bond network. So there is a penalty in the scoring function for buried and satisfied polatom. And the last step is also done with Rosetta with a motif graft mover, where the motif is then grafted on the scaffold protein, and the scaffold is also refined similarly as for the seed, so that the scaffold has a better match with the protein target.
Awesome. Another question that came in said, I'd like to ask if there are any possibilities for subsequent server deployments or easier use of your code lineup. It doesn't feel like it's very easy to update in various dependencies at the moment.
That's true, because MaSIF-Flu is using a lot of things, so it would be also for us much easier to make it a bit more user-friendly, because, well, when things change on the cluster, it's always annoying when we have to recompile everything. But this is still a task that no one was very courageous to try so far. What I can tell you to make your life easier for now is that we do have a Docker container on the GitHub so that you can easily run it locally on your computer without installing one by one all the different softwares and tools that we're using. So just use a Docker container. It will just make your life much easier.
Awesome. Another question was, do the seeds have to be contiguous or non-contiguous sequences in a patch? Are the fingerprint databases available? And how is the scaffold searched for graphing? So it's kind of a couple of questions, so we can take it
Yeah,
one by one.
I'm just like going step by step. So for the first one, well, if the seed have to be continuous or non-continuous sequence, uh, in a patch, let me see if I understand the question correctly. So in the patch of the target or the seed, because the seed the seed is actually a small fragment. It has different overlapping patches on the surface. Same for the target actually, and well, it's continuous here for the helical motif. It's not necessarily continuous for the beta strands, because beta strands, as you know, they have loops in between, and in the process we are cropping the loops because they are way too difficult to graft on the scaffold protein. So here we focus only on the beta strand of that motif. So when we do the grafting step, if you have a beta strand like this one, it will be discontinuous and grafted discontinuously on the scaffold protein. That's also why the success rate for grafting beta strands is lower than for helical motif. I hope it answered the question. I'm not sure if I got it properly. And are the fingerprint database available, and how is the scaffold search for grafting? So the fingerprint database for the seed is available. Because it's really, really big, we had to made up some solutions. So I think at the moment, the best thing we found so far was to pay some Google Drive so that we have it available. I think now the link is on the GitHub. If not, just email us and we can then forward you the link to download the database. And the scaffold search, well, this one is actually in the data availability statement at the end of the paper. So it's on Zenodo, you can easily download it.
Did you have anything you wanted to follow up on, Rakesh, about that? I saw you
Yeah,
unmuted.
I just had a question, like when I say contiguous or not contiguous, right? So on the interfaces, let's say there are a couple of amino acids which are, let's say, on a particular beta strand, right? And there are another on a particular other beta strand, but now they are not in terms of sequence, in the sequence space, they are not contiguous. They are like in different sections of the sequence. Does your seed database have like those kind of motifs where they are structurally close together, but sequentially they're far apart? And
So
does
you
it?
mean on the target side? So you mean if on the target we are targeting different spots?
Yeah,
Or
it could be both on the target side as well as on the search, right? Like your whatever motifs you have, are they like contiguous or like they don't? By contiguous I mean like in the sequence structurally they are like in the space they are quite close, but sequentially, right, like they could be far apart. They could be like beta strands which are coming together to form a beta sheet, but in sequence space they are quite far apart.
So the seed, the seed that actually bind to the targets, the place on the target, the site on the target is not necessarily continuous. So what we
Okay.
only care is actually the surface in the space. And as you can see here, for instance, on that part, we have different helical motifs, and then we have the seed coming on top, but they're not necessarily continuous. What only matters is how are they represented in the geometric space in terms of proximity in the space, but not necessarily sequentially.
Okay. And the search is basically looking at the embeddings, right? Like you're basically trying to take the fingerprints and look at. how close they are, right?
Yep,
Okay, thanks.
exactly. So you have your patch, it has a certain fingerprint, which is basically a one-dimensional vector. And then on the other hand, you have your seed that also have a one-dimensional vector. And then you just compare the Euclidean distance between each of them. And you try to minimize that distance. And if you have, let's say, the perfect match basically will be to get 0 if you do this one minus the other one. But that never happens in reality, but that would be the ideal case.
Thanks.
Awesome. We had another question that came in from Austin. They said your initial use of AlphaFold 2 for refinement was based off how well that tyrosine side chain matched the crystal structure. Given the variability of modeling rotamer side chains, how much stock do you take in predicted side chain orientations for de novo proteins? Different state-of-the-art tools such as AlphaFold, RosettaFold, Chai1, etc., will likely sample and propose different rotamer orientations.
That's maybe true, but in the end, if you compare with the cursor structure, well, AlphaFold was completely right. So I guess here the truth was AlphaFolds in that case, because I'm not sure if I can show again the crystal structure, but this was like crystal clear that we have a good match. And for sure, I will not consider too much for sidechain placement, but in that case, it was perfect. But the sidechain placement had an influence on the folding of the rest of the protein. So that was important for us, because if this is too off, then you won't get a binding. And I guess you need some featuring method at some point. So you need to fix a criteria. And that's also why we get some RMSD threshold. So, and we are not too strict with them. It depends how many design you want to test. But overall, this is so far one of the most of the best state-of-the-art tool that we can use to do so. But I wouldn't trust it too much for sidechain placement. But for for the core of the protein, most of the time, this is quite clear that this is correct. And if the core of the protein is not placed properly, this is what will make the folding incorrect as well. So yeah, I hope it answered the question.
Austin also added on, assuming you have no a priori structure for screening. What would be your approach with that?
No a priori structure for screening, no structure of the target or the binder, because for the binder, they were all de novo. So for that, we didn't have any. For the target, well, in our case, for the design pipeline, for that specific case of the protein ligand complex, you do need to know where the ligand and the target are in complex together. Otherwise, you're just bringing too many variables and degrees of freedom. So the chance of success and the success rate will be quite low, I'm afraid. So you need to be quite sure of at least something if you want to design the other partner. But for sure, you can, I mean, if you have an AlphaFold structure and you're like super confident about it, you can still run the pipeline. So there is no obstacle with this regarding the pipeline itself. But you need to be sure of at least something, otherwise your success rate would just drop dramatically.
Awesome. Another question we got was, in your design pipeline, do you consider potential side chain conformation changes on the target after binding, specifically?
Initial side chain conformation chain on the target after binding. That's possible, but you mean a change of conformation that is induced by the small molecule or by the other protein partner itself, like the binder that we designed? So we thought about allostery, where for instance you can have a small molecule that induces a change and an allosteric change, and then the binder can come on another side. So that's something possible, but at this by the other protein binder. We didn't look at that, but considering that most of the things are exposed to the solvent, they're like quite versatile and they have a lot of degrees of freedom and a lot of variation in terms of sidechain. It's very likely that there will be some changes and reprogrammation of the sidechain conformation. I didn't look carefully, but now that the structures are available, I guess this is an easy exercise that we could do, but we didn't look at that specifically.
Awesome. Ryan asks, have you evaluated the MaSIF fingerprint quality for AF2 predicted structures versus experimentally verified crystal structures? So I guess kind of going off some of our previous conversation and questions.
Yeah. We didn't do that in terms of fingerprints, because the fingerprint comes like very early in the process. This is something that we could have done at the end as well, just to check that the final designs are actually are still matching the fingerprints that we got at the beginning. So that's something we could do. We didn't do it in that case. Because you start with a lot of designs, and the funnel strategy makes them like lower and lower, and in the end we have so few designs that we prefer to test them all just to be sure that we do not miss anything. But in theory, that could be another featuring method that we can add some kind of retro feedback loop in our process. So in theory, this could be a nice idea.
Awesome. I'm just going to give it a minute to see if there's any more questions. We are getting close to time. Oh, perfect. Another question is, can this pipeline be adapted for designing binders for DNA or RNA?
In theory, it could. The only problem that we have so far is that the pipeline, as it's written in the code, can handle only one ligand or one small molecule at a time. So it will need some little adjustment so that there is more than one non-protein components in the process. Because, well, I don't know if you're familiar with the DNA in the PDB, but it's like a small molecule. It's one of these 3-letter code ligands. So, and each nucleotide has 1-letter code. So if you want to do so, you will need to handle the code a bit differently so that it can accept to have more than one small molecule, let's say, in the protein ligand complex, and compute the different geometric and chemical features. So on the principle, it can, but the code has to be a little bit adapted for that.
Awesome. Well, if there's no more last minute questions, I just want to thank you again, Anthony, for coming and speaking with us today, to the community for coming and asking some insightful questions. And with that, we'll see you guys in a couple weeks for the next one. Thank you guys so much.